let's first say that there is no generality. Every nano object, every nanoparticle could be different. We could say yes for some and no for others. If you're doing a small nanoparticle, like magnetic particle, you can use for MRI contrast agent, then that's pure chemistry. That's really simple. Even though you have to qualify the product manufacturing process and to be sure that from one batch to another, it will be reproducible, but like any other manufacturing process. Now, if you start thinking about a nanoparticle that will be coated with something, plus you will have some monoclonal antibody on it, then you're getting into trouble because that kind of objects are very complicated to manufacture. So that's really depend on the object itself you want to manufacture. But from the core nanoparticle itself, at the end, it's simple compared to biologics because most of the time it's purely chemistry.
I should say that it is also part of the evaluation of the risk associated to the development of each of these potential solutions. And it's also what we try to implement in Europe with the ETPN, is to understand perfectly that for us as a large industry representative, we want to decrease the risk associated to the development of any of our solutions. If we succeed, considering again that the very early research will be in academia, that SME could be the right partner for the innovation, we need in that case to put in place in Europe the adequate infrastructures in order to solve all the bottlenecks. We mentioned the toxicological aspect. We need to leverage that. We need to standardize. We need to put in place some robust way to move forward, but also we need to put in place some dedicated structure who are able to produce industrial, the nanoparticles, which are developed in academia in a very robust way, as Laurent mentioned it, in order to move forward mainly through all the development processes. For us, as an industry, if we need to develop or create a specific tool, industry tool, just to produce some batch for our development plan, it will be certainly a disadvantage for the nano. So we need to see what will be the place of academia, SMEs, support of institutional organizations such as Europe, but it could be done also at the member state level or regional level, just to be sure that we can move these nanoparticles up to first in man, as an example, just to demonstrate that the risk associated to the development of these particles can be handled by us. Otherwise, we are always, and I think that my colleagues can say the same thing, we are always evaluating all type of risk. And if there is a specific huge risk associated to nanoparticle, we are not going to risk nothing, I should say.
I would like to add, yes, every innovation has new risks, but we should not forget the opportunity.
Yeah.
That's the balance you have to find as a business, I guess, and not to, have everyone now leaving this room with the impression there's too much risk connected to nanomedicine or whatever. Just this comment. So I think if the balance is, it can be really a breakthrough. It can open a new therapy. It can open a new patient population or whatever to be treated. Then I guess we have to acknowledge that risks can be acceptable, especially in indications like oncology, where patients will accept that maybe a first application of a new type of treatment has some side effects connected to it because it's simply life-threatening.
To be more precise, the risk I mentioned was mainly the industrial risk, not the risk for the patient. I think that it should be even the opposite. The my consideration about the value of nano is really associated to the benefit for the patient. So we need to identify the adequate segment, the adequate pathology, the adequate strategy of treatment where nano will be a real breakthrough, decreasing, and it is the case in oncology, decreasing the toxicity, because again, the chemistry, I think our colleagues from the pharma can easily say that the chemotherapy, more or less, if I remember well my education at the university, we are trying not to kill totally the patient, but just limit the toxicity to the right dose. So it is another concept. We are not looking at and again, we go back to the real specificity and the value of the nano. We should explore much more the advantage of these particles. Again, the difference between the chemistry where the dose is the effect, in nano, the size is the effect. So we need to see how these particles can be, in oncology as an example, a fantastic tool at each cell level. It is a fantastic breakthrough. We need to look at that. But again, it's very new. There are a lot of challenges to be solved, but it is evident that the numbers of projects which has been developed during the last 5 years, it was more than the growth is more than 500% of growth. There are today more than 250 or something like 267 projects ongoing in development in nano. So it is evident that it is an actuality. It is something done on a daily basis, but we need to put in place the adequate structure, organization, business model to move forward in the more efficient way. But it is evident that there is a fantastic adventure associated with the nano.
Bertrand, you mentioned the toxicity of chemotherapy. First of all, just a point. I think the targeted therapies are not devoid of toxicities. And primarily, when we are considering that, at least today in some indications, cancer is becoming to be a chronic disease, toxicity, even not life-threatening toxicities, will become more and more an issue when you will have to administer treatment not only for months, but for years, like in chronic myelocytic leukemia, for instance, today. And being able to overcome these chronic toxicities by using formulation, nanoparticle-based formulation, can be really a very interesting approach, because clearly we know today that in some circumstances, you cannot stop the treatment. You cannot stop the treatment in renal cell cancer, for instance, when you stop sunitinib within very few weeks, you have a relapse. And definitely, this will become a major issue. And we have an example, because I think that the pegylated interferon is a first example which allowed to give to patients with hepatitis interferon for long term, and we all know that it's something which is very difficult to tolerate when you have to take it for months.